Next, we will examine some photomicrographs to see how arteries, veins, and capillaries appear under the microscope and whether we can identify them. These images help us understand the structural differences between the vessels more clearly. Looking first at the artery, we notice a thick, muscular, and elastic wall, which is clearly visible in the photomicrographs. The lumen, or the internal space through which blood flows, is relatively small and maintains a more circular shape compared to the vein. This structure helps the artery withstand the high pressure of blood pumped directly from the heart. In contrast, the vein has a much thinner wall. Its lumen is generally larger and can appear irregularly shaped or collapsed in tissue sections. The thinner, less rigid walls make veins more flexible but less able to resist high pressure. The key differences are most apparent when comparing wall thickness and lumen shape. Arteries have a much thicker wall relative to their lumen size, while veins have thin walls. Additionally, the artery's lumen is round and well-defined, whereas the vein's lumen is larger, irregular, and sometimes flattened. These features make it possible to distinguish arteries from veins in photomicrographs. Now, if we look at another photomicrograph, we can observe all three types of blood vessels together. Arteries are easily identified by the thick walls and narrow lumens, veins by the thin walls and wide lumens, and capillaries by their extremely small size with walls only 1 cell thick. The structures of arteries, veins, and capillaries show distinct differences under the microscope, reflecting their specific functions in the circulatory system. Arteries have significantly thick walls made up of prominent smooth muscle and elastic tissue layers. This allows them to withstand the high pressure of blood being pumped away from the heart. Their lumen, the internal passageway, is relatively narrow and usually maintains a round shape in cross-section. Arteries are generally larger vessels overall. Veins, in contrast, have much thinner walls with less smooth muscle and elastic tissue. They carry blood back to the heart under lower pressure. Their lumen is typically wider and can appear collapsed or irregular in tissue sections. In terms of overall diameter, veins are generally similar in size to corresponding arteries. Capillaries are the smallest blood vessels, with walls consisting of a single layer of endothelial cells. Their lumen is extremely narrow, often only wide enough for red blood cells to pass through in single file. Due to their tiny size, capillaries may not be easily visible at the same magnification as arteries and veins. So, now, this makes it easier to identify blood vessels in both photomicrographs and diagrams, as the key structural differences are clearly visible. When observing blood vessels, we can distinguish them by examining the relative thickness of their walls and the size and shape of their lumens, which is the internal space through which blood flows. Arteries generally have thick, muscular walls, known as the tunica media, which help regulate blood pressure. Their lumen is usually round and relatively small compared to the overall diameter of the vessel. In photomicrographs, the thick, pink, smooth muscle layer is a prominent and easily recognizable feature. Capillaries are the smallest blood vessels and have the thinnest walls, consisting of a single layer of endothelial cells supported by a basement membrane. They lack the muscular and elastic tissue found in larger vessels. In images, capillaries appear as tiny, thin-walled structures, often just wide enough to allow a few blood cells to pass through in single file. Veins have much thinner walls than arteries and a larger lumen. Their tunica media is less developed, and because of their thinner walls and lower blood pressure, veins often appear collapsed or irregular in shape in tissue sections. These characteristics make veins relatively easy to distinguish from arteries and capillaries in both diagrams and photomicrographs.